Hello?
Yes?
Yeah, I've been calling you for 15 minutes.
So?
15. Yeah, can you believe that? Anyways, I called you for a reason, not to annoy you. It's bio again. It's like DNA replication, right? And it's through one ear and out the other. It doesn't make sense.
To me, that sounds like a skill issue. Well, I'm busy right now, so I'll send you a video to help you.
Yeah, that works.
Well, I gotta work on other stuff now, so bye.
I'll see you later.
Hello, welcome back to another video. We'll be talking about DNA replication. DNA replication occurs during the S phase of interphase in the cell cycle. The two strands of DNA run anti-parallel with one another. One runs 5' to 3', and the other runs 3' to 5'. Let's get a closer look. The nitrogenous bases are bonded with hydrogen bonds. Helicase first binds to DNA and starts to unwind it by breaking the hydrogen bonds between the bases. These hydrogen bonds are quite easy to break in comparison to the covalent bonds along the backbone. Helicase starts at a point of origin and moves outward in both directions, creating a replication bubble. The ends of the replication bubbles, where the helicase is, are called replication forks. When the helicase is doing its function, SSB proteins, or single-stranded binding proteins, bind to the DNA strands to keep them separated. Gyrase is ahead of the helicase and basically situates the DNA and prevents the strands from supercoiling. Next, primase attaches to the two strands and adds RNA nucleotides that act as primers. Primers are needed so that the DNA polymerase III has something to build off of. Both primase and DNA polymerase III build from 3' to 5'. On the topic of DNA polymerase III, this is where it comes in. Its function is to add DNA nucleoside triphosphase to the primer. The DNA bases arrive with their own stored energy, which is used for binding. This energy comes from the 3 phosphates attached to the sugar. When the DNA polymerase III is building the new strands, there is a leading and lagging strand. The leading strand is the strand that is building in the same direction that the helicase is unwinding the DNA. The strand is able to be built continuously. The lagging strand, however, is building away from the helicase. As the new strand is being built, the DNA polymerase III must move from 3' to 5', so it keeps jumping back to where the helicase just was and continues building. This causes the formation of Okazaki fragments. As we can see, the two strands are separated and built off of individually. A new strand is built off of an original or parent strand, making DNA replication semi-conservative. Moving on, DNA ligase then comes in and acts as a glue, patching up the gaps between the Okazaki fragments. After that, DNA polymerase I checks the strands and looks for errors to correct. So, why does replication need to happen in an anti-parallel fashion? Well, this is because the strands are the same structure, so if they go the same direction, then the bases can't bond. Besides that, DNA replication relies heavily on the direction of the strand, such as the building of the leading and lagging strands, and the direction in which primase and DNA polymerase III move in. Well, that's all for today's video. Don't forget to like, subscribe, and hit that notification bell.